Instrument preparation. Power on GE4 to pre-warm before testing. Begin the test when the ready indicator appears. Reagent preparation. Pre-equilibrate all components at room temperature for 10 mins before use. Sample preparation. Immerse the tip of the swab into the sample extraction buffer, rotating 15 times clockwise, then 15 times counterclockwise. Break it at the breakpoint. After screw up the cap, invert it 5 to 8 times to mix. Then let it stand inverted for 3 mins for later use. Loading sample. Add 3 drops of prepared sample to the test assay tube. Wait 30 seconds for the lyophilized reagent to dissolve. Invert the tube 5 to 8 times to mix thoroughly. Then gently shake the tube 2 to 3 times to bring all liquid to the bottom. Insert the test assay into the GE4 device. Analyzing the results. Results will automatically display on screen within approximately 10 min.